Okay, everybody. I'm Rob Burgess. I'm Chief Business Officer for Sinobiological, and I would like to officially welcome you all to the next in the series of Sinobiological hosted webinars called Lock and Key Immunodetection. So I want to officially welcome you all for attending this webinar. We have a great speaker today from the company called IsoPlexis, who I will introduce to you in just a minute. Just a few housekeeping issues, really one primary housekeeping issue, and that is that we are going to ask you all to type your questions, if you have any, into the chat box, and just do this throughout the speaker's talk and seminar. And at the conclusion of the seminar, I will verbally run through all of those questions in the chat box and ask our speaker to basically answer those questions. So that's probably the best way to proceed. So with that being said, we're running a little bit behind schedule, so I'm going to go ahead and introduce our esteemed speaker. And that speaker is Mr. Sam Rivera. Sam is the Senior Sales Manager for the company IsoPlexis, and he has around 20 years of experience in life sciences, biotech, and pharma. I will tell you a little bit more about his background. Specifically, he has received a Bachelor of Applied Science in Biochemistry from Rowan University. And he also has a Bachelor of Applied Science in Biochemistry from Rutgers University. Prior to his position at IsoPlexis, he was a business development manager at Minari Silicon Biosystems. And prior to that, he was a sales manager at Mesoscale Delivery. And before that, he was Director of Business Development at Covalent Development and Director of Sales and Marketing at the Philadelphia-based Lampire Biological Laboratories. So with that, I will officially welcome and introduce Mr. Sam Rivera and turn it over to him. Thank you, Sam.
Thank you, Rob. I'll start by asking if you guys can see the screen that I'm presenting right now with the instruments and my pointer. Excellent, excellent. All right, quite a good introduction. I appreciate it, Rob. I'll take it right from the top. So today what we're going to be talking about is our IsoLight system or the platform for IsoPlexis, the kits that we have. Sorry, let me move this out of the way. There we go. The kits that we have, and also the software that drives the whole system. To give you an idea of what it is, this is what our instruments look like. We have the IsoSpark, which is the smaller of the two instruments, which you can do 4 experiments at a time. And then we have the IsoLight, which is the bigger one, which has up to 8 experiments. Basically, our instruments are this. They are an ELISA processor, an automated ELISA processor, where the reagents from our kits would go either in this tray or drawer or this drawer, depending on the instrument that you have. The instrument is a visualizer in the center, which has not only a CCD camera, but different lenses. A visualizer very similar to many others that are out there in the industry. But where we're very unique and different is that our instrument on tops are actually an incubator. So again, it's an automated ELISA processor, a visualizer, and an incubator all in one. Going back to here. All right, so what does our instrument do? Let me see if I can miniaturize this. So our instrument is actually something that can be used for 3 different applications, completely different applications. Quick question, Rob, are you seeing this bar on the top here in my presentation also? You are, all right. Let me see if I can kind of take that away, because that's maybe distracting.
Well, I see the evolution of functional proteomics. That's what I see as kind of the title of it.
Oh, understood. But you don't see something on top that says mute, start video, etc.
No.
Okay, good. Yeah, it's only on me. Good, because otherwise it would be distracting to the audience. All right, good. So as long as we're clear what we're seeing, I'll continue. So our instrument has 3 specific applications, completely different. They are, everything's based on ELISA, and one of them is the Multiplex bulk ELISA. This is very similar to Mesoscale, to Luminex, to Quanterix. ELISA is something that just about every researcher knows, so I won't get into it. But when I speak about highly multiplex ELISA, I'm talking about those platforms where you can look at several analytes simultaneously from the same sample. And full disclosure, and kind of something that Rob mentioned, I not only worked for mesoscale discovery, but also for Luminex in my career, so I really understand this field very well. And where we stand above and beyond the instruments that are out there is that for this application, for the bulk application of our instrument, we're better in 3 ways. Number one, we miniaturize the assay. So while in my MSD or Luminex assays, you will need hundreds of microliters of sample to do a multiplex, with our platform, you only need 11 microliters for bulk, 11 microliters. The second thing we've done besides miniaturize it is that we automated it. The entire process, where with an MSD assay, where you would have between 4.5 hours to 6 hours of hands-on, including several pieces of equipment before you can take your plate to the reader. With us, you take your biological fluid, you do a simple dilution to the limits of detention of the instrument, you pipette your sample into the chip goes in the box, and you walk away. And the third thing that we've done to improve on these other highly multiplex ELISA platforms is that we have increased the sensitivity by 1 log. How do we manage that? We not only use the best camera and the best digital platform that's available for us for the instrument, but we also got the best monoclonal antibodies available to be able to give us the most sensitive ELISAs. So within that application alone, and anybody here that does bulk, that uses MSD, Luminex, et cetera, you know the pains that go with that and the costs that go with it. With this application alone, if you're using, if you're doing bulk analysis, it would justify our instrument. But that's not all. That's actually sort of one of the side applications. The main application, or the application for which the instrument was invented, was for this: functional immune landscaping. The easiest way I can describe this is single cell functional phenotyping. And these are live cells. Now, the same instrument can also do omics. With the same instrument, a different chip and kit, you can actually find out what's going on inside of the cell using our phosphoprotein-based omics kits. So right off the bat, you see that either one of our instruments, you can run 3 different applications, completely applications, and the only difference is obviously the protocol that you run, but also the chips and the kit that you buy to run those applications. That's why we refer to our instrument as a proteomic hub, or even a Swiss Army knife for your lab, because it has so many different applications. And I can even foreshadow there's a 4th application coming up, which is going to be metabolomics. Because once you know what is present in your sample, which cells are secreting what and by how much, and why they're secreting, the next question asked is usually, okay, what is my cell-to-cell interaction? Or what was the metabolic state of my cells before I did this analysis and did my treatment, et cetera. So that's yet another application that's coming for our instrument in the future, which is going to be metabolomics. And later in my presentation, I'll start to touch a little bit on the kits. But this is why we do it. The reason we do it is, and not only to drive the research that our researchers were doing, before we became IsoPlexis at Yale University, we actually knew and we understand, and 60% of the people, we did a huge survey, and 60% of the researchers in our space told us that they lack meaningful data or robustness to fidelity to function. In other words, they can get a lot of data, but the understanding of how that data is representative of the actual biological functions, there's just not a lot of fidelity to that. Where have we turned to? Where we have turned to recently, and especially since the Human Genome Project was completed, was genomics. Things like RNA-seq, or companies like 10x Genomics that are really focused on the genomic side of the cell. But if there's one thing that I never get any pushback on, is the understanding, even when presenting to genomic teams, is that there's a low correlation between transcription and true cell function. In other words, It is, in other words, within the cell, we know that it may have the ability to do something genotypically, but that does not necessarily mean that it's going to express itself phenotypically. And what we have seen is that only 40% of the time when we find out, yep, these are the proteins that are going to be secreted, only 40% of those that data ties directly to what happened. So that's really a gap there, but it's a gap that has been accepted by the scientific community because there was nothing else. Where what we do that's different and where our value proposition lies really is in what we can do. With different technologies out there today, you can look at different things such as, oh, sorry guys, wrong slide. Oh, there we go. With different technologies, you can look at what type of cells you have. Like a flow cytometer would tell you, yep, this is the type of cell, and this is, and it may even sort them out to you, and if it's gentle enough, maybe give them to you back alive. Or you can do block cytokines, where you're looking inside of the cell to find out what's the cell doing using phosphoprotein blocking block cytokine assays. Or you can look at RNA and DNA sequencing, again, RNA-seq, as well as 10x Genomics and all the other platforms that are out there focusing on that. But what all these things give you are pieces of the puzzle. What they don't give you is exactly what the type of cells that you're studying, what they were secreting, and by how much. Here's another representation of that. But again, with today's technology, we can find out, we can get little pieces of the puzzle, which we later extrapolate back and mostly figure out what was happening with the cell. The value of what IsoPlexis does that nobody else can is that we actually trap live cells. These are cells from your sample, which you have captured, you enrich for, you stain, and you stimulate using our kit, because our stain does not kill the cell. That's very important during a live cell assay. So what we do at IsoPlexis is we capture your live stimulated cell within a chip that is imitating in vivo. We are allowing it to secrete what it would secrete, and that allows you to differentiate within your sample into your cellular assays, which are what we call the superhero cells. Because we all know in our immune system, we have different cells. We have cells that go to the site of an infection and say, uh-oh, there's a problem here. And they not only start to attack the infection, but they start to signal other cells, or in other words, orchestrate the healing process. That's what we call supercells here at IsoPlexis. But what we have also determined and been able to learn, especially in an era of COVID, infectious disease, and things even such as rheumatoid arthritis, where your own body, your own immune system is attacking you, there's also supervillain cells. These supervillain cells actually are the ones that are creating havoc in your body. They're signaling, they're sending mixed signals or bad signals and creating a disease state. What our platform allows you to do is capture live cells, differentiate what type of cell it is, and target either your therapeutic effect to help the superhero cells, or the super cells do their work, or your therapeutic effect may be to block the supervillain cells, so the cells that are really driving the disease process. So going back to that needed metric that we had, is that again, RNA-seq and all RNA-based analysis for genotype only has a 40% correlation. We remember this even from basic science, that a cell's genotype does not always equal its phenotype. And more importantly, how does a cell express itself phenotypically during a normal state versus a disease state? That is the measure that was never there before, because never before have you had the opportunity to, again, capture live cells, which you're enriched for, stain and stimulated within our chip. And because our instrument is an incubator within the 13-hour incubation, where the cells are still alive throughout it, they're secreting what they would, and allows us to get the data and extrapolate out not only which are your responders and your non-responders, but also which may be your super responders, which you'll see here in this finger-like projection, or your supervillain cells, the ones that are driving the disease. This kind of information, this kind of metric is revolutionary because it was never available. Now, I myself, when there's something new, I am reluctant to believe anything right off the bat. So I have quite a few slides here which will address, and they talk about the areas or the disciplines that we are in: cancer immunology, cell therapy, infectious disease, inflammation, and targeted therapies. This is some of the voice of customers, some of the information that we've gotten back or someone say, but the most important part in this is it tells you where our publications are, what our publications are, what you can get, where you can get more information on what we do here. And obviously, if anybody wants to dig a little bit further, I invite you to send me an email and we can get you some very specific papers, but also in our website under scientific resources at IsoPlexis. So you just go to IsoPlexis Scientific Resources and you click on that, and you'll see hundreds of papers that are there for your review in many different disciplines. Many times my prospects or even customers find papers there that are very relative to what they're doing and they can relate to what has been published. Now, let me get into how we do this. I can speak about this freely because our technology is well patented, and of course, so I can definitely share, open the Pandora's box a little bit and show you what's inside with it. So here, this is how our chips work. So again, you guys understand ELISAs, but just in case, you have what is known as your capture antibody, your analyte of interest, whatever that may be, your secondary antibody with a reporter to it, or a sandwich ELISA. What we do is the sandwich ELISA, our chips are based on the ELISA technology. This is how our chips work. The chip, which is roughly the size of a Wrigley's pack of gum, it's not very big, has 12,000 microchambers that run left to right within the chip. Those 12,000 microchambers have 32 barcoded antibody stripes, ELISA stripes that go across or perpendicular to that. So every single microchamber, where here is represented with a cross section, each microchamber has 32 stripes of different analytes that it can pick up and report on. It's very simple. It's very straightforward what we're doing here. The magic really happens in the chip and how we capture the cells, etc. But it's that simple as what you see. Again, a translucent chip, which captures the cells that you put in it, which allows those cells are captured within a microwell. That microwell then is measured using the 32 barcoded antibody stripes for a ELISA base. And that's really how you get your readouts. So here's another representation, but I'm showing you the two types of chips that we have. Sometimes there's confusion here, so I'll draw an imaginary line here and talk only about code plex. Plex, think of multiplex, think of MSD, Luminex, Quanterix. This chip is only for the bulk application, and it competes directly with those companies that I mentioned. This chip is very straightforward. You see here, you have 20 macrowells. Of those macrowells, 4 of them are used for a background reference. The other 16 are so that you can do 8 experiments in replicates. In other words, 2 wells per experiment. Each well only takes 5.5 microliters of volume. So that means that you have, with 11 microliters or 2 wells, you can run an experiment in replicate. Each one of these chips allows you to do up to 8 experiments. It could be the same experiment 8 times. It could be 8 different conditions, 8 different time points, 8 different experiments altogether, whatever you may choose. But again, this is the CodePlex. This is the multiplex chip. Very straightforward, very simple to use in our automated platform. You took your sample, whether it be blood, saliva, cell, media, whatever it may be, you diluted it. You pipetted that dilution into the microwells. The chip goes in the box. You press go. You walk away. You come back 8 hours later to get not only the entire assay ran, but you get interpreted data as well as access to the raw data. The second type of chip is the isocode chip, and basically this is the live cell chips. This is bulk on any kind of biological fluid. These are the chips that we use for live cell. We use it for metabolomics and omics. This is a type of chip where the cells are actually trapped within those microwells and kept alive and kept happy while they secrete, and we measure what may be. Again, a cross-representation of a single cell within a microwell, secreting what it would, just like it would in vivo, and being reported back by the 32 barcoded antibody ELISA stripes that go perpendicular to the chip. Some of the common questions that I get here is, how many cells do I need to go into the chip for my live cell assay, etc. So I'll just walk you in a simplified version of our assay. You took your sample, whatever that sample may be that has the live cells, and you have eluted the live cells, or if it's already in media, suspended, and you enrich for the cells of interest. In this case, CD4, CD8 cells. So you took your sample, you enrich using the Miltenyi Clinic Max magnetic bead kit. Very straightforward. Now there's other ways of enriching. We understand and we know that, and we'll work with you with any other form that you have of enriching for the cells, just to make sure it won't interfere with our kits. But at the end of the day, whichever way that you enrich your cells, you will now, using our kit, both stain and stimulate the cells that are of interest, and you create a suspension of 30 microliters at a concentration of 1,000 cells per microliter. In other words, you take 30,000 cells, which have been suspended in 30 microliters, and you pipette it into this port here. That fluid goes into the chamber, into the chip itself, through Poisson distribution, meaning there's no microfluidics for the cells or anything to get clogged. Basically, you put a fluid into a chamber, and the fluid naturally takes the shape of that chamber. Whatever is suspended in the fluid, in this case cells, they migrate to the bottom of the chip, and they settle into these microwells. On average, of the 30,000 cells that you put in, we only need between 500 and 1,500 cells to meet the criteria, because this is how the instrument works, and this is important. While most other companies, when they create a chip, a cassette, a plate, whatever it may be, and you load it with your sample and it goes into their instrument, the instrument will read the entire chip, cassette, plate, whatever it is, and then report in all of it. It will look at it and say, okay, I have errors here, 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 and it will calculate those out using formulas, etc. What we do is a little bit different. Our instrument will actually scan the entire chip. The instrument will say, okay, this well is empty. I'm going to use that for a background check. This well over here may have 2 or 3 cells. I'm not going to count that well because I don't know what cells go what cells are secreting what. But these microwells here that you see represented do meet the criteria, and that is it a single cell? Yes. Is it only 1 cell per microwell? Yes. Is it alive? Yes. Is it secreting? Yes. Is it alive at the end of the assay? Yes. So all these things is what the instrument measures or looks at, and we only measure the microwells that are actually relevant to the study. That's why the information that you're getting back isn't something that has been corrected by software or some kind of a instrument feature. Why the information that you're getting is the actual information from the live cells or the lyse cells, lysed from your omics, that went into the chip that meet all the criteria. That's why we can have good representative data with as little as 250 cells. On average, we see about 1,000. With 1,500, 2,000 or more, it's also common. But again, you don't need that many cells to meet all the criteria for you to get good statistically sound data. So going back to the bulk chip, the chip that again competes with MSD, Luminex, Quanterix, looking at it, really the important things about it again is that we miniaturize the assay, so you only need 11 microliter per replicate, not hundreds of microliters. This is especially important when you're working with mouse samples, when you have limited samples. when you had samples that were earmarked for something else and you have a little bit left over, 15, 20 microliters, and you're wondering, I want to get more information out of that. So that's always there for you. It's fully quantitative. Look at this range between 2 and 2,000 picograms per mil. That's 1 log better than anything you can have out there, than anything there's out there in the industry. And that is achieved by 2 things. Again, not only our instrument has the most advanced digital platform available. So from the hardware side, we have the best hardware available, but also from the antibody side. Again, we buy the best commercially available monoclonal antibodies for our instrument, because we all know that any instrument that is dependent on biology to function properly, it really is only going to be as good as the biology. And that's what's important here, is that the reason we can get this kind of quantitation and these limits of detection, this kind of consistency, is because we're using the best commercially available antibodies out there, period. So again, with this application, the 3 main things is walk away. So you're going to take your sample, you dilute it, a simple dilution to get into these values, so 2 to 2,000 picograms per mil. You pipette the 11 microliters for 2 wells, and you walk away. You get the best-in-class quantification, and it's completely automated. When you come back, so again, you took your sample, you diluted it, you put it into the chip goes in the box, you walk away. It does the fully automated ELISA, and then the software not only runs your instrument, but also interprets your data, which is important because, and not so much with this, but with the other assays, when things are sent to labs, to bioinformatics labs, sometimes that's a major delay or major cost. Our software not only runs the instrument, but it interprets it for you. However, if you want raw data, that's easily gathered, and I'll show you in a little bit here. Okay. This here is a quick example of how bulk can help with an analysis. This is a paper that came out in Nature in 2017, where basically we had researchers that noted that they had a sample with cells that were metastasized, and they wanted to know if it is the cell or something in the media that's driving metastasis. So they were able to clean out and take nothing but pure media and move that over to cells that were not metastasizing. And lo and behold, they started to metastasize. So the discovery there was, okay, there's definitely something in this media that is driving the cells to metastasize, that's driving this cancer. So using 11 microliters, they were able to run a panel and look at all the usual suspects. But specifically here, they were able to see that, wait a minute, it's IL-6 and IL-8 that are highly elevated, which are some of the usual suspects that actually drive the disease. So this was something that was quick and dirty with very little sample, but they were able to get information that was very helpful to them. Now, speaking about the single cell secretome, or the single cell chips. The single cell chips, which again, a different application, that's why the chip looks different, but it allows you to look at up to 32 cytokines at the same time. The fully automated ELISA workflow, now mind you, you do have your normal healthy cells that you use and that you're cultivating and that you've taken care of. Of course, you know, that's your sample. So you have taken your sample, you enrich, stain, and stimulating using our kit, and then it goes into the chip. So after you put that sample into the chip, the rest of the assay is automated. It's very consistent, 15.8 CV on it, less than 20%. And again, the sensitivity, a function of not just the instrument, but the actual analytes that we use. Now, everything that you see here that says coming soon, actually this one, the previous one had coming soon, has been released already. This is the best slide that I can show to tell you that At this time, when you go to our website and you just click list of panels to see what we have available, you see that we have all these panels for the bulk, for the cytokine, bulk, chemokine, etc. These are the different panels that we have. Each panel has different analytes, obviously. For more details on that, you can ask me or visit the website. All of our kits are always targeting for human first. Then we add mouse, then we add non-human primate as other species. We're adding more and more of those every day. For the panels for the bulk, we have all these, including one omics and one metabolomics. In other words, it gives you information in these disciplines as well using our panels. We have the panels for the live cell assays, which include all these guys and these different species. And then our newest application, which is the omics and the metabolomics, which is what you see in orange. We have more of these panels that are being released every day. So this was our original application. And we had actually, it was an end user that said, I believe you guys could also do bulk. And we ran with his idea and created this. And within these things in the scientific consortium, actually somebody brought out, can you guys do omics? So at the end of the day, because it is an ELISA-based platform, as long as we can create a chip and we can create a panel, this is an open platform. We can keep adding more and more disciplines to look into and more areas to look into moving forward. So again, one instrument, 3, soon to be 4 applications. Here you see one of the single cell chips or the IsoCode chips being loaded. Here it is being read, it's being read by the instrument. We're saying, okay, these are the wells that I'm gonna measure, but it's all done by the software. It doesn't matter whether it is your phone, a new car, any appliance, anything out there, or any device for your lab. In my opinion, they're only as good as the software. They're only as good as their usability. And that's where I think that our IsoSpeak software really stands out. Like I mentioned before, the software not only runs your assay, but it also interprets the data. Here, what you're looking at is once your assay has been run and you're looking at your data on your workstation that comes with the instrument, or on your computer, on your laptop or another computer to which you have loaded the IsoSpeak software. Once you have the data, then you can look at it within the software. You can turn on and off any analytes you want by simply clicking on it, or you can look at things like which are secreting, all, etc. You can look at different donors or donor groups or conditions. All of this with a simple click of the mouse. So once your experiment is run, you can look at it in so many different ways as far as conditions, etc., and analyze, but also how that data is interpreted. You want to look at it as a heat map. You want to see it as a compound column with further information, or a 3D Tisney. That's all available. But for those who really want their data, who want raw data, exactly how the instrument read it, when it read it, what cell, etc., you have this. You have your export feature, where you simply put a USB into the instrument, you press export, and you get the raw data files, the CSV, exactly how the instrument read them. So you can have the best of both worlds. Again, once your experiment is run, you can click on it. You can see it as a 3D Tisney, responder, non-responder, etc. This is really neat. You can rotate it on its axis. Or you can see it as a heat map. Or you can see it as a PAT-PCA. Or you click and you can look at it as a compound column. The 3D Tisney and the compound column are actually two of the most popular ways that our customers look at the data, because here it not only tells you what's present and how much, but really if it's an effector, stimulatory, chemoattractive, regulatory, inflammatory, etc. So for example, if you're doing your work and you're looking for inflammation markers, you're going to take a glance at this immediately. You're going to say, oh yeah, inflammation there, there, and there. Those are my 3 big inflammation. I want to know more. You click on it. Now this compound column graph becomes a simple graph where every single analyte is shown and the concentration. And it allows you to actually group your experiments in a folder. And you could do things such as pre and post treatment, and even post treatment, what actually moved. So that's something that, again, very easy to use, all click of the button. This is a reiteration of the same, but in the CodePlex. So this is how you look at your polyfunctionality or your single cell data. This is how you can see your bulk data, which is, again, you can look at a compound or simple columns, etc. And this is actually the most common way, just for a quick snap, where customers look at to see, okay, this is what's being secreted. These are the different conditions or patients, and these were the time points, etc. So that's really there. Now, something that excites me about the software, and that I think is something, quite honestly, that we're all going to see in the scientific community moving forward, is more of an internal QC of an instrument. For us, the IsoSpeaks and the IsoSpeaks software, as well as the instrument, have something called the IsoQ score. The IsoQ score, basically what it is a quality control score that takes in consideration many parameters. How many quality cells you had, were the cells alive, did the cells secrete throughout it? What else? Were the walls sealed? Was the cell alive at the very end when the experiment terminated? A lot of things go into the IsoQ score. But the beauty of it, that if you as a researcher, you have an aha moment, you have a discovery moment using our platform, the first thing we tell you is run back to your experiment and look at your IsoQ score. If it's red or yellow for some reason, it means that you could be seeing ghosts and it needs to be optimized. And at that point, you can work with our FAS and FSC team to find out, how can I optimize that experiment, or what criteria wasn't met? Why isn't it? But if you have a discovery and you run back and you have a high IsoQ score, you can take that data to the bank. You have had a breakthrough. And that tells you that not only what you're seeing is real, but that you're going to be able to repeat it time and again. This is a feature of our software and our instrument, but quite honestly, I think that we're going to see more and more companies, as they move into the digital era, to have these kind of internal QCs, which is going to give confidence to the end user. So anytime that you have something that is disruptive or revolutionary, absolutely different, you meet the skeptics. I myself, I can't say that I'm not. I am a skeptic as well with everything. And I was a skeptic about this technology and this company before coming here, and I really challenged it. So what I have in the next few slides, really, it's the different institutions in the different disciplines and the voice of customer, what they have seen, what they find that is a complete breakthrough for them, or the abilities that they have in cancer immunology, cell therapy, and COVID-19. And again, our publications are not white papers that our marketing team puts out there. Our publications are done by our customers and institutions like these, in disciplines such as these, in major impact magazines like Nature, Stem Cell, Blood Advances. I mean, we have plenty. So for the skeptics, we just have slide after slide after slide after slide of examples of what it is that we're doing with Caltech, with ISB and UCLA, and the voice of the customer and what it is that they're doing. For this, if this the presentation, or if our breakthrough technology seems too good to be true, it means that I got your attention. It means that you could have a use for this. I say I'm more than welcome. I'll take any questions today or any challenges, because when it comes to providing data, providing actual publications, giving you guys complete visibility into what we do and how we do it, IsoPlexis is like no other company that I work with. We really are transparent, and it is the reason why we have so many publications. This one on cancer cell, for example, in my opinion, a little heavy-handed to say, we're kicking genomic profiling to the curb, really meaning, why would you spend thousands of dollars using 10x genomics, RNA-seq, or other technologies, knowing that genomic profiling is only going to give you 40% of the correlation between what it's actually doing? So here, the thought was, now that you can measure actual proteins coming from a cell, why would you do this? I know it was premature, and I think that it's something that that may come to fruition in the future, but as of right now, you just know that we are really getting all the attention by basically being able to do things like no other companies. Here's an example of a single cell treatment in a bispecific antibody treatment for non-Hodgkin's lymphoma. And basically, what we're saying here is, the pharmacodynamic effects and the immune correlations or responses to the biospecific antibody mastunabab was released of refractory non-Hodgkin's lymphoma. What this allows Genentech to do is, IsoPlexis is the only technology that can predict what cells, in other words, we can tease out what are the supercells, we know how the supercells are working, and we know how, and that gives you an idea how you can better target your therapeutic approach to those cells. Because once you know what are the difference between your responders and your non-responders, you can focus on your responders, and within your responders, you're going to find out exactly how it is that they're doing their thing to allow you to be able to support that. Here is a study in combination therapy from MD Anderson. Another area that we really broken into is infectious disease. We were doing some work before COVID, really, with HIV, with rheumatoid arthritis and the other immunodiseases. But since the age of COVID, as you know, it's been a really, really heavy focus on that. And this one is basically a functional monocytes in COVID-19 reaction in a study that was done by Merck and the ISD Consortium out of Seattle, Washington, where basically they were able to take samples from patients and say, okay, these are healthy patients, these are non-ICU patients, and these are ICU patients. In other words, the ones that required serious help because they were under stress. And with this study, they were able to say, okay, we're seeing a lot more activity with the CD4 cells, and within the ICU patients, to blow that up, we see that there's inflammatory, regulatory, chemoattractive, and effector. We know that these type of cells or supervillain cells are driving the disease, and this is how they're driving it. And that's how they were able to differentiate between both, then move into a treatment regimen using tocilizumab, which shows actually the overall polyfunctional strength of the cell. Polyfunctionality, or PSI, or polyfunctional strength index, is the measurement that we use to tell you how those super cells or supervillain cells are acting. It is a very simple metric. What it means is, within your sample, how many of those cells were either superhero super cells, or in scientific terms, how many of those cells were polysecretors, and those polysecretors, what they were secreting. So that gives you so the polyfunctional strength index is, the formula is how many polysecretors you had in your sample multiplied by the actual analytes or cytokines, so they're polysecreting, gives you a polyfunctional strength index. It's that straightforward. That's all that is. Again, how many cells are polysecretors and what they're secreting. So, and PSI within the single cell application does have predictive capabilities, because in this study, as you can see here, the researchers knew on day one, based on the polyfunctional strength index, that they knew day one that most likely CD, CU, and CUD are going to be the therapies that are going to be most helpful to within their sample. But in order to prove it, to test it, they still have to do 58-day clinical. And while doing it, lo and behold, while all the other conditions fell off, at the end, the ISO-targeted treatments, in other words, the ones that had targeted those polyfunctional cells, were the ones that had the predictability that allowed them to say, okay, if we focus on these cells, they're going to be the ones that are going to give the most results. This really brings me to the story of how our company came to be. This is how IsoPlexis came to be. In the mid, I think it was 2014, it was, IsoPlexis, not IsoPlexis, but Yale and Merck were working together in HIV drug. That HIV drug looked really good and very promising until it got into clinicals where it failed miserably. At that point, the PI from Merck turned to our now our inventor, to Dr. Ron Fan from Yale, and said, Ron, if we only knew then what we know today about these cells, we would have never gone down this path and wasted all this time and money. A light bulb goes off in Dr. Ron Fan's mind. He goes back to the lab at Yale and he creates, using well-established basic scientific principles, you know, cell therapy principles and ELISA readouts. Using those principles, he created a workflow. Once he had that workflow, then IsoPlexis opened to the public to do this as a service. Hey, send us your cells. We can give you predictive information. We can give you a lot of really cool data that you can't get anywhere else. What happens then is that we got absolutely flooded with orders. And at that point, there was a decision to be made. Either we get a huge warehouse and pack it with technicians and do a whole lot of CRO-like work, or do we go to the next level? So at that point, Yale speaks with some of their contacts at Stanford University, and they say, hey, guys, we have this protocol that works very well for polyfunctional strength indicators for cell analysis. Can you invent the machine that can actually do all this? And that's what Yale did. That's what Yale and Stanford did together. They invented first the IsoLight, the big instrument. Since the inception of this instrument in Q3 of 2018, we have sold over 150 instruments, either IsoLights and IsoSpark, and we continue to grow because, again, when you have an instrument that can do that entire process internally, that is an automated ELISA processor, visualizer, and a incubator all in one, it has a lot of capabilities. And we continue to add more applications to it. This is how IsoPlexis came to be today. We think that we're going to continue to grow and add, we know we're going to add more applications and more species, but we're also very malleable in a company that listens to our customers and really our prospects and our scientific board to see what else can we do with this proteomic hub that we have created. That's it for now. I will take any questions or any comments at this point.
Great. Thank you very much, Sam. That was an excellent seminar. We really appreciate your time today. Now I'm going to immediately turn to the chat box and start running through some of these awesome questions that we got. I'll start with technical questions, and then maybe I'll get into a little more about the company and the business.
Very good.
Yeah, the first question we have here is from Shagan Mujahid. Forgive me if I mispronounced that name. And the question is, from the beginning of the experiment to result, approximately how much time does it take?
That's a good question. So it depends on A, how many chips you're running through the instrument at one time, and B, which type of chips. So let me break it down. The simple chip is the bulk chip, where you're taking your biological fluid, you diluted it to what you needed, to the concentration that you need. You put that chip in the box. You have invested about 15 to 30 minutes in doing a simple dilution. Then the chip goes in the box. You will have readouts from one chip in as little as 6.5 hours, and that is a complete run of your assay and data analysis. And then if it's 2 chips, it goes up to 8.5 hours, 3 chips, etc. So it really depends how many chips, but per chip on that run. Now, when we're talking about the single cell experiments, those take longer because there is a 13-hour incubation where the cells are actually kept alive during the actual assay. So if you were to run one single cell chip from the moment that you press go to the moment that you come back and you have fully interpreted data, is 16.5 hours. If you do 3 or 4, you're about 24 hours, but no more than 26 hours to run all of the chips in that application at one time. Does that
still really quick for live in-cell data, right?
Exactly. Most of the time so remember, when you're using the single cell, you're getting about 32,000 data points on average per chip. So the software not only ran your assay for you, but it's interpreted the data and it gave it to you. And that's a good point, Rob, because many times with many assays, when you get raw data, you have to send it to a bioinformatics team or use your bioinformatics software. I have heard of sometimes weeks going by before they get a result. With us, you're going to have it. You're going to have an interpreted result at the end of the actual assay.
Great, great.
Mm-hmm.
Question from Sutini Nichadaman. Are all the panels for blood only? I'm not sure if maybe you.
No, actually. So the panels for the bulk is any biological fluid, blood, saliva, sputum, any biological fluid that contains the analytes that you're looking at. Obviously, if you're looking at something like sputum, for example, you're gonna have to dilute it for viscosity, and that's something that we can discuss with an FAS. But no, any biological fluid that has the actual analytes that you're looking at is the one that you can use the instrument for.
Great. A question from Kyle Mixon, who had several good questions. This is the first one. What about identifying and elucidating post-translational modifications? Do you have that capability?
Post-translation modifications?
Yeah, like phosphorylation of a particular protein by a cell, for example.
Yes. So that would be either the omics or the metabolomics kits.
Mhm.
Now, mind you, the kits that we have, we don't have as many as we have with the other applications because we're getting launched on that. So most of the time, the conversation starts with, do you have the capability? Yes. Then the second part is, do you have my analytes? If we do, great. If not, we can customize a chip for you, or we will look to see in our pipeline how close we are, because we have a lot of analytes in our pipeline which have not been fully validated yet. But when we get information from the field saying, or customers saying, hey, these 3 analytes are really important to us, we can get it prioritized. So when it comes to the actual analytes, if you don't see it, don't worry. Talk to us. Let's see if it's something that's going to happen soon. But we do have within the omics and metabolomics kits, those actually look at phosphorylated proteins, and you can do before, after treatment, etc.
Right. And you had mentioned you started out doing services and then transitioned into an instrument and consumable sales company. But are you still doing contract work? And this is a question from Ahor Terlecki.
The answer is yes. So we still do contract work, and it's really work that it's going to take the next level of expertise, or we're going to work with you, or really you don't have a lab and you don't have a wet lab to do this kind of work. So we can do work internally for you as a kind of like a CRO. We can also put you in contact with some of our CROs that we have in the area that could do the work for you. But we also have something called an IsoPace program. The IsoPace program is something where it takes time, and honestly, it's not cheap. It's it's somewhat costly, but it is because we're doing the research with you. We're going to develop a new method for you. We're going to optimize it. We're going to validate it. So there is even options for us to work for you to take advantage of our level of expertise with our instrument and our technology, but with your theories or whatever it is that you're working on. There's more than one avenue to do that. However, we have transitioned to an instrument company, and whenever we talk to a lab that we think, hey, it'd be easy enough to get an instrument in here and train some people and have you do it yourself and tweak it, or make your own discoveries on the spot, we work on that. We give that priority, quite honestly, but we don't turn anything down. And whether it is in-house or through a CRO, if you have samples that you want me to run for you, we'll find a way.
Right, right. And I'm asking one of my own questions. I know a lot of people have this though. What's the prices on your instruments and your consumable chips?
Okay, that is a good question. So the big boy, or the IsoLight, the first instrument that we have, it lists for 263 all inclusive. But I don't know who I'm talking to, and to give you guys full transparency on what we do. That one goes into pharmaceutical, into pharma, biopharma, etc., for about 170 all inclusive after discounts and considerations, and it goes into academia for 155.8. Academia gets the best price on everything because we all benefit from academia doing better, but also we understand that as a company, those are grassroots efforts. The more people in academia touch our instrument and run our chips, the more then we'll go into the workforce and say, hey, I want an IsoLight because I want to do XYZ with my samples.
Right. And I have a question from Nigeria. Do you sell into that country as well?
Into Nigeria, yes, we do through our ROW, rest of the world. Rob, I realize I didn't fully answer the question about the cost, and I think that's a very important one. So if I may, let me go back to it. So with the bigger instrument, we sell it between 155 and 170, depending on whether you're in academia or not. For the smaller instrument, which lists the 150, we sell it between 95 and 110, again, depending on whether or not you're academia. Academia and GSA gets the best pricing. On the chips, the consumable chip that I mentioned, let me see if I have one available. I do not, I apologize. But the consumable chip that I mentioned that does the bulk, that is 8 experiments in one. Those chips are $350 for 8 bulk experiments. The chips that I mentioned that, I'm sorry, and with those bulk experiments, you get 8 times 32 is 256 data points that you get out of one chip of bulk. And again, you can throw just about any biological fluid that has the cytokines of interest. The chip that we use for the single cell, those are a little bit more expensive. They're about $530 for the chip. When I say the chip, by the way, I mean the chip, the kit, and everything that comes with it. Everything that you need besides your sample is in the kit. Everything that you need except for your sample. And that's it. That's really both types of chips and also both types of instruments. We know, quite honestly, that the biggest hurdle we have is the purchase of the instrument. Because once the instrument is in place, whether it's in someone's lab or in a core lab or a CRO, the cost of the chips and per experiments is very reasonable, and that's where we get more activity throughout, through repeat business, really.
Right. That's a lot of data points for roughly $350 to $500, not including the instrument cost. So that's fantastic.
Yeah. And remember, the software interpreted that data for you, so you're not, you're not going crazy with 20,000 data points.
That's great. And Irina Sokolchik is asking, how do you confirm that the cells are still alive at the end of the experiment, or do you do that?
So as the instrument is measuring throughout it, there's stimulation and there's it's part of the IsoQ score. So what it'll do is it will know if it was stimulated, if it was secreting, and if it stopped secreting. The cells are alive until the end of the experiment, at which time when we do the clean and disinfect everything has been disinfected and killed at that point. However, we have had customers have come back to us and say, hey, after all this is said and done, is there any way that we can have those cells back? The answer has always been no. Now the answer is maybe. Let us work on another development. Let's see what the future brings. But quite honestly, we're working toward a way to giving you your cells back even after you have done the experiment for you to do anything else that you may want afterwards.
Right. That's great. And a couple of business-related questions. Again, these are from Kyle Mixon. The first is, what does your intellectual property portfolio look like, and like how many patents do you have?
No idea. That's one question. Honestly, that's one question that I know that we're heavily patent, and I know that as a salesperson or senior manager that has such insight into it, most companies are very careful about what they tell us, because the reality is that we want to talk about it to the world. So here we have we have been told and trained well to talk about everything with lots of transparency, knowing that we have a lot of IP behind us to protect us. If
Right.
you really want to get like detail or more detail, you can either go to our website and say about us, or again, I'm Rob, I'm hoping that you're distributing my email to everyone. Send me any question from anywhere in the world and I will answer it or put you in contact with the person. And I'm going to look that up. I want to know basically how many patents we have for the next time I get asked that
Yeah.
question.
Was your email on your last thank you slide? I didn't notice.
Um, I'm not sure, but
Okay.
I'll reiterate it real quick for you. It's what we're using. It's my name, Sam.Rivera
Mhm.
at isoplexis.com. That's it.
I just want to make sure you get some leads out of this. I mean, that's a good opportunity for you to get your email out there. So,
No, absolutely. Again, Sam.Rivera at isoplexis.com. Send me any question, technical or not, or even curious. I'm here at your service for that.
what about cynobiological antibodies and reagents for you? Are they working out well for you?
They are actually. When I mentioned earlier about finding the best commercial monoclonal antibodies, we mean it, and that is something that while working at MSD and Luminous, God forbid I mentioned that we got antibodies from other companies, but we do. We're transparent about that because we're not an antibody company. We don't have a farm and we don't create our own. So, and we know that the rubber meets the road with all these technologies that uses biology. The rubber meets the road with the actual biological reagents that you use. And so we source nothing but the best available. And that's the root of the relationship that we have with Biological.
That's great. We really appreciate that. And thank you for the kudos on the quality of our products as well.
Well, we're in it together. And I think
That's right.
we wouldn't be
Partners.
this good without you guys. Otherwise, it would just be another so-so platform. So
We do appreciate that. So
mhm.
I think that's a good way to end the seminar/webinar. And first, let me thank everyone who attended. We had a huge turnout today. We appreciate all of you staying for the entire seminar and for the Q&A. And I want to thank Kyle and the others who attended and asked some really, really good questions in the chat box. We appreciate that. Made for some good discussion. Certainly, Sam, I want to thank you for an excellent seminar and for accepting our invitation to give this seminar today. Kudos to you and the entire IsoPlexis team. I feel like y'all are really on to something, and it's just a wonderful organization that you work for. And then, I just want to personally thank my colleague, Dr. Jen Liu, who's our marketing manager and is the one behind the scenes who actually executed this cynobiological hosted webinar. So with that, I want to thank everybody once again, and I'll adjourn the webinar, and everybody have a great day or a great evening. Thank you.
Thank you, Rob. Thank you, Dr. Liu. You guys have a great evening as well.
Thank you. Bye-bye.